Thank you for joining us today. My name is Kübra, and I am a global marketing specialist from IBM Technology. We are happy that so many people are attending today's webinar, so big thanks to scientist.com, our distributors and business partners who promoted this event around the world. Today's presentation will be about the latest technology in live animal imaging, real-time intravital microscopy by Cayus Professor and IBM's Technology CEO, expert presenter Dr. Pilhan Kim. During the webinar, if you have any questions, please use the Q&A box below. We will also answer some of the questions sent prior to the webinar. Dr. Pilhan Kim, you may start with your presentation. Could you please mute? You're unmuted.
Okay, sure. Okay, thank you for the introduction, and then thank you everyone for joining today's webinar. So today I'd like to introduce our technology called real-time intravital microscopy. It's capable of in vivo live animal imaging in cellular level. Okay, so this is the one of the representative data the intravital microscopy can provide. So compared to the tissue level imaging modality like X-ray, CT, or MRI, the intravital microscopy can provide a very detailed live video in cellular level in a live animal model. So in the middle, this is the video. It's actually 10-minute video obtained from the lung of sepsis mouse model. In green, we can see the blood circulation, basically blood flow. And then every red dot in here is actually the neutrophil. That's the one of the major type of the leukocyte inside the blood vessel. And all this red dot is a single individual neutrophil. And then you can see in the middle, this neutrophil, they contact together, and then suddenly it completely block one of the vessel here. Yes, now, and then this blood flow is completely stopped. So this is the one of the major cellular level event that happened in the lung of sepsis animal model. Okay, to give you some idea, so this is how we do it. So we make a special device for the lung, stabilized lung imaging. So in the middle, we expose the lung of this mouse model. And then by suck out the air through this hole in here, we can build up the negative air pressure inside this chamber covered by transparent cover glass. And then by doing that, we can stabilize the lung of the live mouse model. So by this, after this stabilization, we can take this high resolution microscopic imaging images from the lung of this mouse model. Yeah, we can apply same principle to other organ like heart. So same principle, we can, this is the our device for heart imaging, stabilized heart imaging. And then this is the real-time video obtained from this setting. So and then this red dot is actually the red blood cell inside the vessel of this heart. Okay, so and then you can see this flowing red blood cell inside the vessel of the heart of this mouse model. So actually the heart is still beating, actively beating. We just stabilize the just a small portion of the heart, and then we can acquire this real-time video in cellular level. So we can monitor, we can track the flow of the individual single red blood cell and also lysin positive leukocyte inside the vessel. Well, not just the cell, we can also image the drug actually after fluorescence labeling. So this is the 24-hour image obtained from the mouse model. And then in this image data, the green dot is actually the cancer cell. It's a breast cancer, human breast cancer cell. And then, so what we do was we make a breast cancer model. using this mouse. And then we implanted this docile skin fold imaging chamber in the back skin of this mouse model. And then in the middle, we injected this breast cancer cell and then let it grow for 2 weeks. So this breast tumor is get big. After that, what we did was we labeled the blood vessel using antibody, Pro4. It's an anti-CD31 antibody conjugate with the near-infrared Pro4. And then we injected this antibody conjugate, and then it systemically labeled all the blood vessel. And then this green is actually the cancer cell. And then if you look at carefully, this red dot is actually the nanoparticle labeled by the red Pro4. So at 2 hours after the intravenous injection of this nanoparticle, it first appears mainly inside the vessel. At 6 hours, we can see just a small portion of this nanoparticle, the red fluorescence nanoparticle, get out from the blood vessel, like this here and here. And then we can also see the increase of this nanoparticle. And then finally, at 24 hours after the intravenous injection of this nanoparticle, we can see a lot of this nanoparticle get out from the vessel and delivered to the cancer cell. So this is one of the very good example how we can use intravital microscopy technique to monitor the drug delivery to the target cell in the live animal model. Okay, that was just several examples. So the intravital microscopy compared to this macroscopic scale imaging, intravital microscopy enables a dynamic 3D imaging of various cellular level dynamics in a live mouse model. So we can monitor cell trafficking, basically cell movement, or cell to cell or cell to microenvironment interaction inside the live model. So these are the examples. So all this cell movement can be directly imaged by using intravital microscopy. And then also for the drug development as well, after process labeling, we can monitor the delivery of the drug in the live mouse model. In the middle, this is the example I already explained. On the right side, this is the real-time video obtained from the liver after the intravenous injection of this nanoparticle. So you can see the appearance of this flowing nanoparticle inside the liver vessel, liver sinusoidal vessel. And on the left-hand side, this red dot is actually T cell. It's so basically what we're looking at here is adaptive transferred T cell targeting the cancer cell in the mouse model. So you can see these 2 T cell, after the intravenous injection of this T cell, get into the tumor cell, get into the tumor site, and they interact with the cancer cells like this. So we can monitor the cell therapeutics as well, in addition to this nanoparticle or other antibody drugs or peptide. So for drug development, this intravital microscopy can enable a direct imaging analysis of drug delivery to target tissue and cell. Also, we can monitor the drug efficacy. And then by doing that, we can validate the mode of action of this drug candidate in a live preclinical animal model in vivo. And then our intravital microscopy solution have been applied to almost every organ and tissue. So, so as I showed you in several examples in the beginning of this webinar, we can obtain a dynamic live cell imaging in all different internal organs, like bone marrow, brain tumor, and then also retina inside the eye, skin, hair follicle in the skin, and then various tumors. Actually, this is colorectal cancer model. And pancreas, spleen, and also gastrointestinal GI tract, like the small intestine, colon, or stomach. And they're all different in adipose tissue, in the subcutaneous or internal, visceral pad prostate, and placenta in a pregnant mouse model was all different, and then different lymph node, and then kidney, well, skeletal muscle, liver, of course, lung and heart, thyroid and trachea in the neck area. Well, I cannot, put every images in here, but we can monitor. So, but basically, we can image almost every organ and tissue in a live mouse model in vivo after some surgical preparations. So I like to introduce our newest model. This is IBM CMS3 model. It's a dual mode, intravital confocal and two-photon microscope. It's very much optimized system for in vivo cellular level intravital imaging of a live vivo model. And then it has all-in-one single box packaging for easy installation and then cost and space saving, and also to provide high usability for common biologists and then the beginner users. And then also integrate all the lasers inside. So it provides hands-free maintenance function of the two-photon laser system with minimal long-term cost. And then it fully integrated with the high-speed laser scanner for high-speed real-time imaging from 30 frames per second to 100 frames per second. So basically 30 images per second or up to 100 images per second with 512 by 512 pixels per images. And then also it's integrated with a very compact 920 nanometer femtosecond pulse laser module for two-photon imaging. Also with integrated 4 detectors for two-photon imaging. And then also it can be connected with the 5-coupled 4 wavelengths laser module for confocal imaging. And then for the confocal imaging as well, we can also include 4 detectors. And then additional laser, such as tunable femtosecond pulse laser system, can be connected through additional optical ports. It's in the behind of the backside of this box. And then we are proud to, to integrate automatic live tissue motion compensation function inside. And then additionally, we provide a touchpad tablet for live animal body temperature monitoring and maintenance functions. And then we also provide all different sets of apparatus for internal organ imaging, like imaging windows. Okay, I'd like to show you some short video to introduce this dual mode intravital microscopy system. So it's all-in-one, single box system, and then it has a sliding door in front, and then we have an animal stage inside. So yeah, so we can prefer the animal model for the intravital imaging like that, and then we can put it inside the box, and then make the focus, and then close the door for confocal and two-photon imagings. Like this. So inside we integrated, both of the confocal excitation system, and then 4 detectors for the confocal, and then also put the two-photon. This is the two-photon laser inside, and then also we can, we include the 4 detector, additional 4 detector for two-photon imaging as well. So yeah, this is the, for example, the 3D imaging of the various neurons in the blood, in the brain, and this is our software, and this is the real photo of our dual mode intravital microscope system. Okay, and this is, let me show you some of our 4-color two-photon imaging data obtained from various tissue using our 920 nanometer femtosecond pulse lasers. So here, in white color, you can see the collagen with this SHG mode. And then in green, it's a nuclei. And then in red, it's actually membrane teratomator expressed on all different cells. And then in blue, this is the blood vessel. And actually, fluorescence angiography. So we intravenously injected advanced blue, and then that can be simultaneously monitored. And then this data was obtained from the live anesthetized mouse model. So this is the skin, and this is the G-stack data. So we change the focus inside the skin of this anesthetized mouse model. And then on the left-hand side, you can see this is the kind of representative maximum intensity projection imaging. So you can see all these 4 colors data. This is a muscle. Again, so this is the you can see in white color. You can see the myocyte, the muscle cell, and the nucleus. This is the pancreas. Again, nucleus, and the cell membrane and blood vessel can be simultaneously imaged. This is the kidney, so you can distinguish individual tubule and the nuclei of cell. And also blue color, you can see the blood flow in between these tubules, in kidney tubules. This is spleen. Okay, so now, this is how we implement ultra fast laser scanning. We use rotating polygon scanner instead of the conventional resonant Guyver scanners. So this polygon enables ultra high speed, like up to 100 frames per second with 512 by 512 pixels. It also provides additional advantage over conventional Guyver scanner because it provides uniform excitation, illumination over entire field of view. And then let me briefly explain about the motion compensation functions. So if you think about it, although mouse is anesthetized during this in vivo imaging, still the mouse heart is beating to pump the blood circulation. And then also it should breathe air, so lung is expanding and close, and then shrinks to breathe air. So there is physiological motion always. And in microscopic scale, in cellular level, all this physiological motion of the heart and lung cause the tissue motions like this. So to deal with this these motions, first we need the video rate imaging capability. So this is a video rate imaging data obtained at 30 frames per second. And then we do frame by frame motion compensation. So by comparing the subsequent frames, we can calculate how much movement was there between each individual frames. And then we automatically compensate these motions. So if you look at it here, frame number 1, 2, 3, 4, you can see this same object, same vessel, it moves around like this. And then we just correct it every frame by frame. And then we can stabilize the motion and then get this high SNR image data. So this motion compensation is essential to get this high resolution image data from moving tissue in a live mouse model. So without this motion compensation functions and the high speed imaging capability, you will always end up with blurred images. And this is another example. This is actually data obtained from the kidney. So you can definitely see the advantage of this motion compensation. And this motion compensation is implemented to be automatic. So this is the video capture of our the software. So on the left-hand side, this is the real-time data. And then what you need to do is just activate the motion compensation function and then select the which color, which detector to use to detect the calculate the motion. And then just single click of this capture button. Then you can get this compensated images within seconds. Also, we provide in vivo live MMO monitoring and maintenance function. So we can monitor the body temperature during the imaging using rectal probe. And then we can provide feedback heater control functions to keep the body temperature to be stable during the imaging. This is quite important for long-term imaging, like several hours, even for 24 hours. And then we also provide additional temperature sensor to monitor the local tissue temperature, and then also cover glass heating system to keep the temperature to be stable during the all the time of the imagings. And then in front we have a sliding doors, and then we have this stage sliding mechanics inside. So we can just take out the staging system, and then we can plate the detachable, plate heater, and then we just set up the mouse with all different apparatus to hold the tissue and mouse at the position, and then we put it back under the microscope, under the microscope objective lens, and then we can close the door. And then we also provide our own intravital image processing software for 2D, 3D, and 4D imaging data. So this is one example of the 3D data set we obtained from the small intestine. So you can, this is all single villi of the small intestine. And then this is 4D data. So basically we obtain the 3D data and then in a time-lapse manner. Then we can see the movement of the individual cell in 3D over time. So this is the 4D, the 3D data, reconstructed data, and then played in a video. And then we recently developed another technique for real-time video rate cellular level imaging data processing. So on the right-hand side, this is a real-time video rate video obtained from the skin. And then this is the typical noise level. So when we increase the imaging speed, we will always have this noise issues. And then we developed the AI functions, AI processing model for noise reduction. So on the left-hand side, this is the real-time video after this noise reduction processing. So you can clearly see the flow of the red blood cell in the single capillary in the skin. And then green is actually the nuclei. And this is the same data set. This is the raw data, raw 30 frames per second video data. On the left-hand side, after the processing. So we can see the flow of the red blood cell together with the muscle, together with the myocyte in the muscle and the nucleus. And this is the kidney data. So all this real-time data video can be processed to reduce the noise level like this. Okay, and so I told you this is a dual mode system of confocal and two-photon imaging capability. So when you compare confocal and two-photon imaging capability, it has several differences. So confocal microscopy is single photon excitations, and its imaging depth, typical imaging depth is different from various organs and tissues, but typically in the range of 100 micrometer. But this confocal microscopy imaging mode is very easy and efficient for multicolor 3D intravital imaging of various tissue and organs. In comparison, two-photon microscopy, two-photon imaging mode utilize the two-photon excitation principle, and then its imaging depth is a little further, so it can be several hundred micrometer in the various tissue and organs. So it's better it's a better imaging mode for deeper tissue, 3D imagings. But for two-photon microscope, we only have one wavelength, so multicolor imaging is a little bit limited. So the selection of the flow for multicolor imaging is limited in two-photon microscope imaging mode. So, well, I don't want to go too much detail about the principle of confocal microscopy, but basically the confocal microscopy is capable of full color fluorescence imaging with the sectioning capability. So we can basically, so without the make a slice, this whole live tissue can be, imaged in sections in a non-invasive manner. So this is our typical data set. So this is the data obtained from the inguinal body tissues. So you can see this three-color video data, three-color image data with the 4 micrometer step size. So you can see the typical resolution we can achieve by confocal imaging mode is submicron in XY directions. So about 0.6 to 0.5 micrometer in XY directions. And in Z-axis directions, in axial resolution is about the 2 micrometer. So you can see, well, so we can basically have this microscopic scale, 3D structure. Multicolor 3D structure can be visualized by using confocal imaging mode in a live mouse model in Vivo. Now for two-photon microscopy, so we use femtosecond pulse laser in a near-infrared, near-infrared wavelength region. And then in addition to fluorescence imaging, it is also capable of label-free non-linear multi-image generation imaging like second image generations, SHG. And then this SHG imaging mode is particularly useful for fiber optic collagen imaging because it doesn't need the process labeling for second image generation imaging. So this is one example obtained from the liver fibrosis model. So all this green is collagen without visualized by the two-photon imaging mode with the second image generation signal. Okay, so these are the example of the second image generation imaging together with the fluorescence. So on the left-hand side, this red signal is actually the RFP signal expressed in the vessel and the hair follicle, and then sebaceous gland in the skin. And this blue color is actually the collagen imaged by two-photon imaging mode with the second image generation signal. So in the middle is the liver fibrosis model. On the right-hand side, this is the muscle. And this red signal is actually a sarcomere inside the myocyte. So this is a dual mode image, the representative dual mode imaging data. This is a wide area images. Basically, we did the TIE scan and then make this large mosaic imaging data. And in this particular example, we imaged the transgenic mouse, the Scion YFP. So this peripheral nerve express yellow fluorescence protein. And then blood vessel was labeled by using antibody conjugate and then shown and then pseudo-colored in blue. And then this is the 3D data with the 5 micrometer step size. In red color, this is the sarcomere, and then also collagen in the fascia of the muscle. And then in green, this is the peripheral nerve. And then actually it's the neuromuscular junctions. And this is the maximal intense projection images. So we can clearly see the blood vessel in blue in confocal imaging mode. And then also this green neuromuscular junction. And then we can switch to two-photon imaging mode and then take this second image generation signal in two-photon imaging mode. Okay, so from now on, I'd like to show you, several examples of live imaging data obtained from the different organ in live mouse model. So this is the lung, I already explained. And then in the lung, since we can do video rate imaging, we can take this kind of real-time data of IPC flows in the capillary of the lung using a transgenic mouse model. So this is the real-time data. And then later, we can do the motion compensation processing and also overlay of the real-time, IBC flow data, and then analyze the individual IBC flows in a single capillary in the lung. And this is the 2-hour video showing the extravasation of lymphocyte like a T cell and B cell inside the lymph node. So we make the skin incision and expose the subcutaneous lymph node like this. And then we can image the different cell, red colored T cell, green colored B cell. And then this blue color is actually perivascular FICs. And then you can see from in the middle, you can see the big vessel. And then from inside of the vessel, you can see the, extravasation of the cell. So this green cell and red cell to get out from the vessel to the lymph node parenchyma through several junctional sites marked by these arrows. And then this is the heart. So I already showed that we can directly monitor the blood flow in the beating heart using this heart imaging chamber. And then this is actually the data of the epicardial pad accumulation after high fat diet. So this white color is actually lipid. It's labeled by using some special flow for SF44. So the intravenous injection of this SF44 systemically label all the lipid inside the body, and then we can take an image, take intravital image from the heart, and then we can detect this epicardial pad in the heart after this high fat diet. And this is the preparation for the small intestine and mesentery imaging. So what we did was we anesthetize the mouse, and then we extrude the small portion of the small intestine. And then using transgenic mouse model, the Proxon GFP mouse model, we can see the lymphatic vessel in green with the GFP signals. And then what we did was we put the red color fatty acid, and then we can see the movement of this lymphatic vessel in the small intestine. And this fatty acid was observed and then transported through the lymphatic vessel from the villi in the small intestine, also through the mesenteric lymphatic vessel. If you get here, after the application of this red pressed fatty acid on the surface of the small intestine, we can see the transport absorption of this fatty acid to the single villi, and then transport through the lymphatic vessel in the mesentery. You can see it's 90 minutes after the application of this fatty acid on the intestinal surface, small intestinal surface. And then, well, that was a relatively short observation, but the beauty of the intravital microscopy is the mouse is alive during the imaging. And then after the imaging, we can let the mouse to recover from the anesthesia, and then we can image the same mouse again at later time point. So this is one good example. So this is the everyday images obtained from the same mouse after bone marrow transplantation. So this red color is actually transplanted bone marrow hematopoietic stem and progenitor cells. And then you can see, from day 1 to day 4, we can see the engraft of this transplanted bone marrow cell inside the bone marrow of the skull. And then from day 3 to day 5, we can see this very dramatic increase of this transplanted cell. So it means active proliferation of the transplanted cell, happen at this time point at day 3. And this is actually 5 hours timeless data. So you can see the active proliferation of this transplanted bone marrow cells in the bone marrow. And then this is the liver imaging. So I already explained, so I just go on. So, well, in the liver, what we can do was, this is just one example. We can monitor the development, accumulation of the lipid droplet. And then this lipid droplet was labeled by this SF44 dye. And then this is actually 3 weeks images, like 21 days of this special diet induce, development of this lipid droplet inside the liver. So these 2 days. And then this is 7 days after this diet. You can see the increase of this lipid droplet in the liver. So this is 2 days, 7 days, 14 days, and 21 days. You can clearly see the increase of the lipid droplet in the liver. And then also, as I explained before, we can monitor the collagen. So basically, we can monitor the liver fibrosis together with this lipid droplet accumulation. So this is the comparison: the normal mouse, normal healthy mouse, and then also this liver fibrosis and fatty liver induced mouse model. So the red color is the lipid droplet, and green color is the collagen increased in the liver. And obviously, we can make it 3D. So from now on, let me briefly introduce the imaging chamber things. So I don't want to explain too much here, but this is one of our educational video uploaded on the YouTube. So we can make this dorsal skin fold chamber implanted on the dorsal skin, and then we can inject the cancer cell at the subcutaneous tissue in the middle of this imaging window, and then we can monitor that this cancer tumor model grows over several weeks. And then in this setting, we can, because we can image the blood flow, we can do the video rate imaging. We can directly monitor the microcirculation, the blood flow inside the tumor vessel. And then also we can monitor the drug, anti-cancer, anti-tumor drug delivery, like for 24 hours, for example. This, and then also we can monitor the change of the same vessel with 3 days interval. This day 7, day 10, day 13, and you can see that this increase of a green cancer cell, and then this increase of and the change of this blood vessel. So you can see this huge dilation of the blood vessel after the infiltration and proliferation of this GFP expressing green cancer cell. And then there's another windows tools like abdominal imaging windows. So basically we can image all abdominal organ like liver, kidney, or pancreas using this imaging window device. And then all this imaging window device is actually modified and then optimized to be used with our microscope system. So this is actually the preparation of the pancreas for long-term imaging. And then, so this is the one representative data. So we implant this abdominal imaging window for pancreas, and then we inject the pancreatic cancer cell, PANCO2, GFP. And then we can see, this is actually the same area at day 9 and day 14. So you can see the increase, the active proliferation of this GFP expressing pancreatic cancer cell, also change of the blood vessel. Well, actually, we can monitor every day. So from day 9 to day 14. So you can see the very dramatic, increased proliferation of this cancer cell, and then also vessel changes. And there's another technique called cranial imaging window. So basically, we remove the small portion of the skull and then expose the brain, and they seal the area with the transparent glass like this, and then UV epoxy. So after this setting, we can monitor the same area of the brain for several months. This is one example study. So what we can do was in here, we can target just one vessel here, one penetrating arteriole in the brain, and then we induce the thrombosis here in this vessel. So basically, we can make a small micro-infarction and then induce mini stroke in this mouse model. After that, we can monitor the same area for 30 days, like for 1 month. And then we can see how individual cell and the tissue was changed over time. So actually, this is the observation, the quantification of this volume of this area. So you can see this is the edema from day 1 to day 2. You can see the very significant edema after the stroke, micro-infarctions. And then later, we can see the tissue shrinkage, basically the cortical atrophy in microscopic scale. And then also we can see, we can test the BBB function. So what we did was we inject the small molecular weight dextran, like a 3 kilodalton dextran, labeled with the fluoro, and then we can see the leakage of this processed dextran. So this is the before, and this is after the induction of the stroke. So you can see this red signal, red fluoro, was actually leaked out from the blood vessel. It should not happen in healthy condition. So this is before the induction of the this microinfarction. After this stroke induction, we can see the transient dysfunction of the BBB. But later at day 5, the edema was reduced, and then at the same time the BBB function was recovered. And then at the same time, at this time period of day 3, we can see the recovery of the pericyte and vessel coverage. So this is actually 24 hours movie. And you can see this red color is the pericyte and blue color is the vessel. So you can see that this coverage of the pericyte over the vessel was recovered for this one day. Okay, so it's almost time. So I just showed a few examples what it can do with our in vivo live cell imaging using intravital microscopy technique. So if you're interested, please visit the website of this iBeam technology. It's www.ibeamtech.com. So you can see all the information about our all-in-one intravital imaging platform. And then for your information, we also, in addition to standard model, we have customized the model as well. So for other, some extreme application for large animal, we can provide this pre-space system, customizable system as well, on demand. And then we provide a lot of example about the intravital imaging of various disease model. And then we also have a webinar series. It's all uploaded in our website. So you can find out the specific application you're interested in, and then you can find the webinar there. And then we also provide research service because we have our in-house animal facility and then in-house lab. So if needed, we can test your material or your digital model. So we provide the imaging plan and then do the test imaging. And then in our in-house lab, we perform all the animal experiment together with the in vivo imaging and then provide a report. And this was one exemplary project published in Science Advances in 2020. So in this case, the company sent us the exosome. And then what it did was we monitored the 30-minute drug delivery monitoring in the sepsis mouse model. This red color is the exosome. So this is the 10 minutes after the exosome injection. We can see the delivery of this exosome to the surface of the target cell. And at 30 minutes, you can see the internalization of this exosome. Okay, so this iBeam technology, we provide this all-in-one intravital microscopy system in the dual mode intravital microscopy system of confocal and two-photon imagings. And then we also provide the imaging service. And then with our in-house specialist team, with know-how of the intravital imaging with more than 10 years experience, so we can provide in-depth consulting and also technical support for optimizing intravital imaging experiment. And then we also provide the training or also technical service as well. So thank you very much for your attention. And then I'm happy to answer any questions you have.
Thank you very much for the great presentation. And we will now take some time for questions.
Okay, so let me answer the question in the Q&A.
Sure.
So the first question is this iBeam only used for animal? Yeah, well, at this moment, this system is designed for animal mostly, but we are in the development of another system for human use as well. But it's not available. It's not, we didn't release it yet, but we are preparing another system, more specialized system applicable for the human. And then the next question is, for subcutaneous tumor model, do we need the user chamber or the light penetration strong enough to make it through the mouse skin? Okay, well, for soft cancer tumor model, we need to use the chamber, the dosage-capable chamber, because both of the confocal and two-photon image mode, it cannot penetrate the full depth of the skin. The typical penetration is 100 micrometer to 150 micrometer in confocal imaging mode, and then 250 to 500 micrometer in two-photon imaging mode. So that's the kind of the depth limit. So we cannot penetrate the skin. So for subcancer tumor model imaging, we need the imaging chamber. And then the next question is, can we track exosome traveling from one tissue to another? Well, the answer is yes, but we need to design the experiment carefully, because at one time point, we can image only one organ. So we can image the release of the exosome in one tissue, and then we move to other tissue to see whether those released exosome was delivered to another organs. But we cannot image the same organ at the same time, but it should be sequential images. Observe the one organ and observe other organ next. And then the next question is, so which microscope technique I'm used to capture the 3D dynamic video? Well, it's confocal and two-photon microscope, basically, with motion compensation and then functions and then all different apparatuses. And then G-stack, yeah, it's G-stack taking with the video recording. And then so try the G-stack image with the recording to generate the 3D dynamic video before, but the cell moved too fast. I was not able to capture the cell to the 3D dynamic videos. Okay, so in this case, what can do was our imaging speed is 30 frames per second. So what we can do was we just take a video while changing the 3D, changing the depths. So we take a 3D video, changing their depths, and then later we can do the AI processing to reduce the noise in the video. And then by doing that, we can later build up the 3D data set in high speed. So like this, So although we are not changing the depth in here, but we can actively change the depth using piezo stage for the objective lens. And then later we can do the AI processing and then reconstruct the 3D data. And then the next question is, can you image awake mouse in this setup? Well, in our standard model, it's difficult. But as I told you, we have a pre-space system. And then in using that pre-space custom system, we can image the awake mouse. And then actually we provide that customizable custom system for neuroscientists before. So the answer is yes, but we need custom design system for awake mouse imaging. Okay, and then estimate the learning curve time for someone with a little experience with the microscopy. Well, it depends, of course. Our software is quite easy to use. So like only 1 or 2 day training will be enough to, control all the software functions. And then this software and our microscope is kind of ready to use. So everything is inside, and then you need to learn how to handle the animal for the first time, and then that will take like several weeks. And then the microscope itself is quite easy to use. Everything is almost everything is automated, and then just single button operations. So I would say, several weeks. And then the next question is, can you image nerves and vessel in the skin without a window? What's the penetration depth? Oh, yes, of course. So we can easily image the nerve and vessel in the skin. So actually, well, this data was obtained from the skin, and then so without a window, and then you can easily see, we can see the vessel with the labeling. And then also, we need a transient mouse or a nerve staining, but we can easily image the nerve and vessel in the skin without the imaging window. And then in the skin, the penetration is about 70 micrometer in confocal imaging mode, and then 150 micrometer to 200 micrometer in two-photon imaging mode. And then the next question is, can you image, can the system accommodate latch? Yes, it can. So it can deal with the mouse and latch. And the next question was the same. Yeah, our system can deal with that. And then the next question is, can animal be on a ventilator during the imaging session? Yes, it can. So we have it's not shown in here, but we have this tube adapter on the side. So we can connect the gas anesthesia system and also ventilator through this tube here. And also we have a tube adapter here as well. So we can, connect the ventilator. We can connect the ventilator or gas anesthetizer through this hole. So we can use ventilator during the intravital imaging. And then, is it always necessary to implant the observational window? No, not really. So in many examples, so if we need to image just one organ at a time, then the imaging window is not necessary for that. The reason why we need the observation window is to find the same location again and again. So like pancreas, for example. Should we use? So to find the exact same location at different time point, like for this example, this is a 3-day interval. Okay, so we image it and then let the mouse recover, and then we try to find the same vessel at 3 days after. To do that, we definitely need this imaging window. So without an imaging window, every time we need to make a surgical surgical procedure to expose the tissue. And then every surgical procedure will be different. So it's difficult to find the same location without the imaging window. And then the next question is, I am working in asthma field. How this machine can be used? Well, in asthma case, we can image the trachea and then also the lung. So and then with the immune cell. So there might be the applications you can use the intravital microscopy for asthma study. And then the next question is, I'd like to ask, what kind of software do you use to take images and to analyze them? Well, we use our own software. So we developed our own, acquisition software, and then also analysis software. But so we use our own software to acquire the image. And then for analysis, you can use other third-party software as well. We provide our own visualization software, IBM Studio, but you can easily use other software like Imaris for analysis. And then, can we use this for studying iPSC cardiomyocyte? Well, yes, you can, because we can image the heart, and then also all other different muscles as well. And then, how deep into the brain can we visualize it? Well, in the brain, it also depends, but using two-photon imaging mode, in the brain, we can normally image up to 500 micrometer. It depends on how much laser power you put in. But typically, it's relatively easy to image up to 300 to 500 micrometer. But when we crank up the laser power more, we can even go further, like 600 to 700 micrometer into the brain. So, in the cortex, almost the layer 4 or 5 neuron can be visualized using two-photon imaging mode. And then how long can the animal survive with the chamber implanted? Well, it can survive, between 2 weeks to even several months with the imaging chamber, with the implanted imaging chamber. Well, I introduced several different imaging chamber techniques, but for dosage pulse chamber, it normally survives like 3 to 4 weeks. And for abdominal imaging window, it normally survives 2 to 3 weeks. And then the cranial imaging window for the brain, it can survive several months, like 3 months to 6 months. So for imaging over days, how to treat the animal to get the same imaging site? Well, with the imaging window, it's relatively straightforward to find the same location. So after one imaging session, we let the mouse to recover, and then at a later time point, we bring the mouse back, and then using our window mounting system to locate the imaging window at the exactly same location and same position under the objective lens. And then we can, just search around it, because our system is 30 frames per second. It's a video rate system. So it's relatively easy to observe such the wide area, such the large area, and then to find the same location. Please provide the e-certificate for this webinar. Yes, I think we can do that later. Then what anesthetic are you using? Can you use isoflurane? Oh, yes, we can. We use both of gas anesthesia like isoflurane, and then we also use injectable anesthesia as well, like ketamine. And then, is the cranium the only bone tissue amenable to I-beam for bone marrow? Well, the answer is no. Well, the cranium is easy. That's why we normally use cranium to image the bone marrow. But if you want to image the bone marrow in a long bone or tibia, then it's possible, but you need additional surgical procedure. But there are many publications already about the bone marrow imaging, the long bone marrow imaging using two-photon microscope. So we can, just follow that published papers method. And then, can you image rodent brain? Yes, we can. I already show you many examples, I think. The brain of the mouse and also brain of the leg. And then the next question is just wondering how the animals are kept under anesthesia while imaging. Well, with the gas anesthesia like isoflurane, well, we just provide isoflurane, and then the mouse will be anesthetized for like several hours as long as we provide isoflurane. And for injectable anesthesia like ketamine, we first inject induction dose, and then it's normally enough to keep the mouse to be anesthetized for 1 hour. And then after 1 hour, we normally use half dose to extend the anesthesia. So yeah, isoflurane can be used. And then, okay, so can isoflurane be used? Yes, we can use isoflurane. If so, is the anesthetic unit coming with the imaging unit, yeah, if you request it, we can do that. We can provide the anesthetization unit together with our microscope. But if you already have an anesthetization system, then we can help you. We can support you to connect the existing system to our microscope. And can an injury be introduced real time while imaging in order to see the platelet aggregation after stenosis in mouse model? Yeah, yes, we can. So using the so each option. So we have another option for laser injury. So you can target the single vessel with the laser, with high power laser, and then induce the injury. And then we can exactly do these things. We can see the platelet aggregation after inducing this laser induced injuries in the vessel. Can you do drug biodistribution study with your instrument? Oh, yes, we can. Yeah, so as I showed you before, so we can, image we can monitor the distribution of the drugs in various tissue and organs like liver or tumor or kidney or other organs. But we need to, fluorescently label the drugs to do that. So are there any issue with the tissue growth that will block the imaging window? Well, not really. So it doesn't happen in many cases. So the answer is generally no. Okay, I think I answered all the questions in the Q&A window. Okay, Sabina? So Sabina raised the hand. So Kubra, can you let her to ask questions?
Sure. Oh, I think she also
Okay.
asked in the Q&A box. So.
Okay, I see. I think maybe I answered her question in the Q&A window.
Yeah. So it looks like we have covered all the bases. The webinar will be available on demand on insightscientific.com. As IBM Technology, we offer hands-on and virtual demos, free imaging test experiments with your samples, and training for animal surgery models. For any inquiries, please contact us at the email information@ibmtech.com. Thanks again for joining us today, and we will see you all next time around.
Okay, thank you.